Welcome, everyone. Thank you so much for joining us today for today's Public Health Discovery seminar. Don't let the mosquitoes bite, careers in vector biology. Today I have with us our lovely Dr. Sarah Michael. She is a faculty member in our Tropical Medicine and Infectious Disease Department, as well as LeShawn Simplis-Barnes, our lovely Director of Admissions here. Just some really quick housekeeping tips. If you have any questions while the seminar is going on with our panelists, please drop them in our Q&A. We will answer all questions with the last 10 minutes of today's seminar, and I will kick it off to Lee.
Awesome. Thank you so much, Kayla. I'm so excited for this panel today, for this seminar. So I'm going to get right into it. And as Kayla said, I am Lee Simplis-Barnes. I'm the Director of Admissions here. And I want to get started with asking the panelists to tell us about your current position, a little bit about what you do. And if you can mention the year that you graduated from here at Tulane WSPH. I think I see Kevin first on my screen, so if you want to get us started and then you can popcorn it to the next person.
Sure, thank you. My name is Kevin Caillouet. I'm a Tulane graduate, first with a master's degree in 2001, and then with a PhD from tropical medicine in 2007. And I realized when they asked for pictures that I don't have very many pictures of myself, except really goofy ones like this one, trying on a mosquito trap here. So I'm the executive director for St. Tammany Parish Mosquito Abatement, just across the lake from New Orleans, of course, only about 45, 50 minutes away. But we have a lot of different mosquito problems here, a big diversity of mosquito habitat as well. That structures a lot of our problems. Lee, would you like me to go through my whole spiel here with this? And I have one more slide too, or are we just going to go around the room?
No, that's fine. That was
Okay.
great. We're just going to do introductions, then
Great.
we can get into the meat. Thank you, Kevin. Appreciate it. Megan, would you like to go next?
Sure. Good morning or afternoon, everyone. My name is Megan Saunders, and I am a senior public health biologist with the State of California's Department of Public Health. And as a quick mention, views heard today are my own and do not necessarily represent those of the state of California or my employer. My job is I coordinate the environmental tick surveillance program for the state of California. So we have over 40 species of ticks in California, about 6 which generally cause problems for humans. So I coordinate testing, surveillance for those, as well as deal with, as a public health biologist, I cover 6 counties in California for anything vector or rodent-borne related. So I work with plague, hantavirus, Chagas disease, invasive Aedes. If it bites humans or spreads sort of zoonotic or vector-borne diseases, that is what I do.
Awesome. Thank you, Megan. Next, if we can have Catherine introduce herself.
Thank you, Lee and Sarah, for inviting me to this panel. My name is Catherine Prusynski. I am a research biologist at the Florida Keys Mosquito Control District, and I graduated from Tulane with my MSPH in 2010, and that happens to be the same year that there was a dengue outbreak in Key West, the first locally acquired dengue outbreak in Florida in more than 70 years. So I was hired to develop a PCR lab to test mosquitoes for dengue virus there. However, we realized that testing mosquitoes after the fact was sort of reactive. So my goal while here is to prevent transmission before it occurs. And I did that with my dissertation at the University of Florida on vectorial capacity. And my current work focuses on Aedes aegypti surveillance, as well as evaluating different control strategies for Aedes aegypti and different population suppression tools.
Amazing. Thank you, Catherine. Next up, Brian.
Good afternoon. My name is Brian Byrd. I'm a faculty member at Western Carolina University, about an hour west of Asheville, near the Great Smoky Mountains National Park. I finished my MSPH at Tulane in 2002 and my PhD in 2009. My job is mostly teaching and undergraduate research. I teach an environmental health program where I teach courses like epidemiologic methods and medical entomology.
Thank you, Brian. And last but not least, John.
Hello, everyone. I graduated in 2006. I was in a PhD program, but also a medical degree at the same time. And so my specialty is the immune reactions to arthropod antigens, and this is particularly covering venom-induced anaphylaxis and the asthmatic reactions that you get from cockroaches.
Awesome. Thank you. Let's get into it, Dr. Michaels. I will segue over to you if you want to introduce yourself to everyone.
Great. Sorry about the struggle with the slides. It's that video, Kevin. We'll work on it. Let's go ahead and start with Catherine. Catherine, you mentioned that you're based in the Florida Keys and you've done extensive work on the dengue vector Aedes aegypti. Could you tell us a bit more about different control and prevention methods that you've been involved with there?
I can. Okay, so at the Florida Keys Mosquito Control District, we use this integrated vector management approach to mosquito control, especially Aedes aegypti control. So that approach combines surveillance, source reduction, biological control, targeted adult control, new suppression strategies, and public outreach. So all of those different strategies are used in our aegypti control program. So surveillance, we set traps, weekly monitoring Aedes aegypti. During my dissertation, my PhD program, I studied the vectorial capacity, which is kind of a look at how many mosquitoes are needed to maintain virus circulation in a population. So I used that surveillance data to determine operational thresholds with this operational goal in mind to develop this transmission threshold. So we use that now, and along with source reduction and outreach, and then additional tools like BTI and adult control when those thresholds are reached, all of that combined helps us keep that population low. Now, we also have these unique challenges in the Keys, right? These delicate coastal ecosystem, a tourism-driven economy, and then our Aedes aegypti population has this permethrin resistance as well. And permethrin is an adulticide that is commonly used for mosquito control. Unfortunately, we can't use it for our Aedes aegypti any longer. So we, in the research department, look at different innovative suppression strategies, right? I'm sure many of you have looked or heard about the genetically modified mosquitoes from Oxitec for population suppression. We tested those along with Oxitec a couple years ago using an experimental use permit provided by the EPA. This summer, we're looking at the Wolbachia infected Aedes aegypti males in conjunction with Mosquito Mate. They've developed that strategy of incompatible insect technique to produce the aegypti population as well. All of this to say, it's a very, very many moving parts of this program, all of which with the goal of what really works in a real mosquito control program.
Thank you, Catherine. Brian, as you mentioned, you're based in North Carolina. Can you tell us more about the work that you've done on La Crosse encephalitis and the Aedes vectors there?
Sure. So, like I mentioned, we're about an hour west of Asheville, and we're in one of about 5 hotspots of La Crosse encephalitis in the U.S. So Western North Carolina, Eastern Tennessee shares sort of a focus, and then in West Virginia, Ohio, and then historically in the upper Midwest. And so every year in Western North Carolina, there's a dozen or more children that end up in the hospital, often with severe infections from La Crosse virus. And if you can see the life cycle there on the left, you see that it's a pretty complex ecology going on, including basically the reliance on vertical transmission or transvarial transmission, keeping the virus in a very small focus. And we've demonstrated where that even stays at a house from year to year to year. And so we've transitioned from a little bit of just focusing on oncology to more epidemiologic work. So we have colleagues at the University of North Carolina Chapel Hill School of Medicine, and then at the local hospital, Mission Hospital. And we have a prospective cohort study where when a child gets sick, our team gets notified. We go to the child's house as soon as possible, do sort of a risk assessment, collecting mosquitoes, collecting serum samples from household members to better understand that risk and opportunities for intervention. So over the last couple of years, we've started to actually focus more on residential interventions, things like addressing tree holes and other types of cryptic habitats around the house.
Thank you. Kevin, you mentioned that you're just north of New Orleans in St. Tammany Parish. Could you share with us some of the work that you've done on West Nile virus, its transmission, especially with Culex and Aedes vectors?
Sure, absolutely. And just as Brian started off talking about his research from the ecological aspect, that's sort of where we started too, and really focusing on birds and that interaction between birds and mosquitoes that are transmitting West Nile virus. And the focus on birds, of course, is complicated by how diverse and wild bird populations are. And each of those different species of birds have different competence levels. That complicates everything. Specifically, wanted to look at the seasonality of the avian nesting cycle and how that is sort of coincidental or contemporaneous with transmission to mosquitoes and how that eventually spills over into humans. So we first started out working on building mosquito traps that could suction mosquitoes off of birds as they're nesting on nest boxes and whatnot, trying to really quantify the rate of feeding onto these birds. We then moved to putting cameras on bird nests, and we got a lot of videos of snakes eating eggs and snakes eating bird chicks and some kind of nasty stuff. But we also got a lot of videos of a lot of mosquitoes feeding on those baby birds too. Those baby birds are sort of the susceptible fuel for the virus every year. And so that's why we tend to see this big pulse in West Nile activity in the summer months, the late summer months after those birds have all hatched and fledged the nests. So we started off looking at those interactions. And then we realized that there's a lot of anthropogenic drivers of mosquito populations around our parish in St. Tammany. If we could go back to that slide with the heavy videos that bogged up the slideshow here, and the one with the funny hat on. Go one more back to the one with the picture of me with, there we go. So this is also another video. If you can click on the video on the right. So in St. Tammany, we have a lot of on-site wastewater treatment systems. These are basically septic systems for an individual house. But the health department allows these to drain into roadside ditches. And this is what it looks like when those systems aren't properly maintained. And at any one point in time, there may be about 14,000 houses that are releasing untreated sewage into the ditches. This sewage will kill the mosquito fish that eat our mosquito larvae. You can see the little dots there are mosquito larvae. So we have a big problem with untreated sewage across our roadside ditches in St. Tammany. And so these are major drivers of mosquito production in our community. They have to be larvicided, of course. We treat 600 miles of this stuff every week in the summer when it heats up like it is now. But also we can try to fight this with some politics and get some ordinances on the books too. So science turns into advocacy, and political advocacy can make changes with changing our community on a healthier level. One step next on that next slide is another video. It's just really trying to kill the PowerPoint here and the Zoom feed. A lot of what I ended up doing is kind of focusing on new applications of technology. How can technology help us improve how we do our mosquito control mission? And so this is an image. The gray image is a LiDAR derived imagery of ground elevation. So we're looking at buckets holding water, and then we're seeing in real time those buckets fill with the hydrologic model that we're creating in-house. And we have those buckets remove water based on the rate of evaporation. We're using game cameras out in the field to try to sync up the model data with what's actually happening in the environment to try to validate our model. But this essentially is a map that our inspectors have in real time on their phones in the field, and they can look and see what areas might be holding water at any one point in time. So I'm really passionate about how do we use GIS, geographic information systems, and new technologies like LiDAR to help us derive more data and information about what's happening in the field so we can be more effective about how we do our mosquito control methods.
you. Thank you. And sorry for the slight delay in playing those videos. Thanks for all of your information. I'm going to try to skip ahead to, let's see, Megan. Megan, you are currently in California, but I know you've worked on ticks everywhere from Maine to California and some places in between. Could you tell us a bit more about what you've learned about ticks and the diseases that they spread?
Yeah, thank you, Sarah. Um, so after I finished my MSPH in tropical medicine in 2011, I moved to Maine to do vector-borne disease epidemiology. Now, what you may not know about Maine is while there are some nasty arboviruses up there, there isn't a lot of arbovirus activity overall. So I became very intimately associated and knowledgeable about tick-borne illnesses because ticks, well, they move slowly. They are the ultimate little hitchhikers. They don't have easily controllable measures the same ways that mosquitoes do in the field. Additionally, they do spread our most common vector-borne disease nationally, which is Lyme disease. So that specifically are black-legged ticks, so our eastern and western black-legged ticks. Now, the other interesting thing about this is, as I said, there aren't a lot of easy control measures for it. So I focus a lot on prevention because the best way to prevent tick-borne illness is to not get bitten by a tick. The majority that cause problems for humans will only feed 3 times in their lifetime. So there's a lot of possibility for intervention from a personal protection standpoint. So with that being said, there are also a lot of differences. In the upper Northeast, close to 100% of black-legged ticks in some areas are infected with the agents that cause Lyme disease, such as aka Borrelia burgdorferi, with plenty of co-infections with Babesia microti, a parasite, as well as Anaplasma phagocytophilum. So a lot of things going on at once with high infection prevalence. Now out in California, what I like to say is we don't have the infection prevalence, but we have a lot more variety. So in California, we have over 8 reportable tick-borne illnesses. So we do have Lyme disease, but our rates of infection are much lower, down at about 2% in adult ticks and between 6 and 12% in nymphal ticks. So the intermediate life stage, which is our riskiest life stage because they're so small and hard to see. So with that being said, we also have all of these other tick-borne illnesses that sometimes get overlooked on the national screen because you don't hear about them quite as much. So we have some rickettsial diseases, which are quite nasty, such as Rocky Mountain spotted fever. There's currently an epidemic along the California, Mexico, Arizona, New Mexico, Mexico borderlands that's being driven by the brown dog tick, which is very interesting because these ticks are highly associated with humans and kennel use. So you can't collect them from the environment in the same way. But that being said, there are also sylvatic or wildland cycles of some of these diseases that are easily detected in environmental samples. So we can go out, do our normal collection methods for those. But once again, that prevention methodology is make sure you don't get bitten by a tick. So what does that mean? Do your tick checks, wash your clothing after coming inside. And certainly in California, there is no non-tick season. There's always something out there, but it does vary spatially between our cooler coastal areas and our southern warmer areas. So there's a lot of diversity, a lot out there. And when thinking about risk prevention, for the most part, you just have to really drive home that message of do not get bitten and be tick aware.
Great. Thank you, Megan. And John, you mentioned, well, we know that you're a practitioner in New Orleans, and you have done some research on cockroach-induced asthma, as well as treating venomous arthropods. Could you tell us a little more about your work there?
Absolutely. So this is work that we've done in New Orleans, really looking at how cockroach exposure within homes leads to poor asthma outcomes for kids in our area. We've found that association to really, the household exposure to cockroaches drives hospitalizations for asthma. And we were interested to determine whether simple, cost-effective interventions would be sufficient to kill off the German cockroaches. That work was successful here in New Orleans, and has now been incorporated into the national asthma guidelines for prevention of and control of asthma. However, in the Gulf South, we have other species of cockroaches that also can be drivers of asthma, but do not necessarily just live within homes. In that middle photo there, you'll see American cockroach on the left, a smoky brown cockroach on the right. And we were concerned that some of these cockroaches would be entering homes, but we didn't know which populations were the ones that would enter homes. So this is just one of the mark release recapture studies we did. That's Kevin Caillouet there, who was gracious enough to pose with a bucket of fluorescent pink cockroaches during our mark release recapture studies. What we found is that most of the American cockroaches that you find in cities like New Orleans, they do live within sewer lines. However, the cockroaches that live within sewer lines don't like to leave the sewer lines, and therefore those were not the cockroaches entering homes. Instead, it was the cockroaches that were out and about, just living in leaf litter and mulch, that would enter the houses. Clearly, they didn't have as good of a life as the sewer line cockroaches, and so they were more apt to travel. So this is just one example of finding out where the populations are coming from that can have a negative effect on human health.
And do you want to tell us a little bit about your work in treating venom-induced anaphylaxis as well?
Absolutely. So for those guidelines, the research was really unclear about where different species of stinging insects are that trigger anaphylaxis. And so conventionally, most of that research was done in the northeastern part of the US. And they identified honeybees, paper wasps, yellow jackets, and aerial hornets as big drivers of anaphylaxis in that region. And so it was presumed that those were the drivers of anaphylactic reactions across the United States, such that if you were stung by an insect in New Orleans, and had an anaphylactic reaction, you would be tested for all of those insects and then desensitized for all of them. However, that recommendation that was out there did not line up with the ecology that we're actually seeing here in New Orleans and elsewhere. And so we initiated a series of studies that ultimately determined that it was really paper wasps driving anaphylactic reactions from stings here in New Orleans. Those are the paper wasps that are, you see the honeybee on that pink flower, right above it are the yellow jacket, the paper wasps that we have in the New Orleans area. We actually collected no yellow jackets in the New Orleans area. Going further afield, you can find yellow jackets, and when stung in other locations far from New Orleans, it is possible that those are drivers. When patients when we can figure out what the species was or the genus of insect was that triggered the anaphylactic reactions, once we get that correctly identified, we can desensitize them to the venom that they're allergic to, so that if they are stung again, they're protected from having a subsequent anaphylactic reaction. We are rewriting the venom allergy guidelines as we speak, and they should be published in 2026, and we'll incorporate this work that we've done in the new guidelines.
Thank you all for your very wide variety and very detailed explanation of the work that you do. Turning it back to Lee to ask some questions about outreach and education.
Thank you, Dr. Michaels. So my first question is for Catherine. Catherine, what has been the most challenging aspect about communicating to professionals and to the public about Aedes aegypti mosquito control measures?
Thanks, Lee. That's a good question. And this seminar is so good. I'm so excited about it. Misconceptions. So in the Keys, we have two primary mosquitoes of concern. We have the salt marsh mosquito, and we have the Aedes aegypti mosquito. The Aedes aegypti is the vector of concern, and then the salt marsh mosquito is the nuisance vector, or the nuisance mosquito. And that's the floodwater mosquito. That's the one that comes off in huge numbers and is really annoying. And there's a lot of misconceptions about the Aedes aegypti mosquito because of that nuisance mosquito. Most people think mosquitoes come from marshes and wetlands, and Aedes aegypti, however, we know has a preferred larval habitat of small containers around homes. She prefers the feeding on humans as well. And then so it's really important then to educate the community and tell them or ask them to eliminate standing water in their yards, right? So mosquito control, we go into yards, but we can't remove every container. And so we have to educate the public as best we can, a lot of outreach events, a lot of going into the schools and asking for their assistance in outreach. And I often think because we haven't had any significant dengue outbreak in a very long time, people kind of take us for granted. So I think there's an old adage, like just like good public health, mosquito control is often invisible, when it works well. So if they don't hear from us, or if they don't see dengue in their community, they think, oh, nothing's going on, but we're doing a lot of work behind the scenes. And that's why we have to keep up with these new technologies, including the genetically modified mosquitoes, which we did receive pushback for. We had a lot of public outreach events to educate and explain what was going on with those genetically modified mosquitoes. And then communication is really important, right? Transparency, listening to those concerns, explaining how and why those decisions are made. And, the key idea here is that we have to have this effective partnership between the mosquito control and the community in order to eliminate disease.
I love that, Catherine. As a former vector control employee, that communication piece is so important. It's so vital to make sure that the public understands the information you're disseminating. So thank you for that. That was great. Same question for you, Brian. However, talk to us about cryptic mosquito breeding habitats, please.
Sure thing. So lacrosse virus is mostly vectored by the eastern treehole mosquito, Aedes tricereatus. And so this is a more sylvan species that will develop in these tree holes that can come in all shapes and sizes. And for most homeowners, they're not really paying attention to tree holes. They've got other things on their mind. And if you look at Catherine's slide here, we still have, the children's toys and tires and buckets and clogged gutters and other sort of, peri-domestic habitat that can easily, a little easier to address at the house. But because we live in a rural area, a lot of these houses where children are getting sick are surrounded by often acres of hardwood forests. So I use the term cryptic habitat here a little bit with a little bit of a grain of salt because as medical entomologists, we know they're out there and we try to find them, but we can't always find those magic tree holes. Some of them are small and some of them are at height and some of them are just, really covered up with other brush and can be difficult to get to. So we're looking at treatment options now that can get different larvicides into these tree holes without even necessarily finding them. And so we're using some different insect growth regulators and investigating potentially the opportunity for some of the mosquitoes themselves to do the work by disseminating like pure proxfen. So that's kind of our main focus on cryptic habitats. It's really tree holes, and they're challenging to work with.
Thank you, Brian. This is just so interesting to me. Okay, so next is same question for Kevin, but talk to us about the importance of surveillance and control programs, please.
Yeah, and also how to communicate that importance to the public is central. And I think Catherine hit it on the head. We can talk till we're blue in the face about, where to find these containers in a yard or how to maintain your septic system, but how do you connect with the public to get that information? What I'm learning is that you have less than one sentence to communicate with somebody these days in terms of that attention span. And look, I wish I could go back to graduate school and learn a lot more about social science and actual about communication. I was all into the infectious diseases that when I was in trop med, but that's what you're already passionate about. Try to, expand your horizons by learning something about, social science and really how to communicate with people. For us, the big challenge for surveillance and control is to get people just to understand, hey, we don't spray on a schedule. You're not going to see the truck every Tuesday night because we have a whole network of traps out there to help us determine where those trucks need to go. It's all data driven. But I've said that for as long as I've been working here, and it doesn't, you're still going to encounter people all the time that think otherwise. So simplifying that message. But again, at the point where a lot of your participants may be, go get the skills that can help you make those convincing arguments and learn from other disciplines to how best to communicate these things that we need to do about vector-borne disease.
Thank you, Kevin. That was excellent. Thank you. Same question, but for Megan, talk to us about tick abundance, distribution, and safe recreation. What does that look like?
Yeah, thank you, Lee. That's a great question. And just to highlight what Kevin said, science communication is so important, but there are a lot of different ways to provide that messaging. I would actually say, so as mentioned in California, there are ticks out throughout the year, and they are very unevenly distributed spatially throughout the state, as are our pathogens. So the messaging that I provide to people in the north coastal areas, where the most common tick is the western black-legged tick, will look somewhat different from that I work with if I'm working in our southern regions, where we have more what we call dog ticks or dermacenter species. Their seasonality is slightly different. They spread different diseases. So a lot of people tend to focus primarily on Lyme disease, which is, as mentioned, the most common vector-borne disease in the United States. It's our most common tick-borne disease in California, but we average about 110 cases a year. So that's not actually a lot. But the big messaging for us, as I said, is don't get bitten by a tick. So when I'm out in the field, that's actually when I do the majority of my education to the public, because they see me with this big white tick flag brushing the vegetation, and they ask me what I'm doing. And the answer is, oh, I'm collecting ticks. So I will brush it, pick it up, show it to them, be like, look, this is what we have going on right now. Now, then that's when that usually goes to, oh, are you testing them for Lyme disease? Well, depends on the species, the time of year, because testing a Dermacenter occidentalis for Borrelia burgdorferi is fairly useless because it's not an efficient vector of the disease. It can't transmit it. However, it can transmit things like Pacific Coast tick fever, Rocky Mountain spotted fever, or our newest pathogen that we just actually identified as a human pathogen in the last 2 years here in my section and with our collaborators in our viral and rickettsial laboratory, Rickettsia lanei, which is very similar to Rocky Mountain spotted fever with some very nasty human consequences such as fever, rash, dry gangrene. It's pretty nasty. So the big thing there is explaining, like, the best thing to do that tick check, make sure you don't get bitten by a tick, because the different pathogens all have different transmission rates. While Lyme disease can take at least 24 hours to be transmitted, something rickettsial can be transmitted within 15 minutes, or a viral disease, say out of a Lone Star tick, which we don't have in California, or out of Ixodes scapularis, such as Powassan virus or Bourbon virus or Heartland virus. These can all be transmitted very, very quickly. So really the goal is to be tick aware, know that they're out there, and don't focus on just one thing. Focus on overall tick prevention, not on the disease itself.
Thank you for that, Megan. Would you mind, because you've talked a little bit about, and I'm going off script here, I know that, but would you talk a little bit about what a tick check looks like and what are ways that you can prevent being bit, right? I know the pants tucked into the socks. You want to delve into that just a little bit for the audience, for those who might not know?
Yeah, of course. So when we're saying do or be tick aware, there are really a couple, or really 4 points that we focus on. First of all, being tick aware, knowing they're out there, which makes you much more likely to do the tick check yourself. Now, a tick check means that when you come in from a potential exposure to a tick infested area, you're going to want to check your body. Now, ticks like warm, moist, hard to reach areas such as armpits, hairline, groin, back, places that are hard to reach. So when you come in, you do a sweep. And I actually recommend that you do a second sweep about 2 hours later, because ticks like to wander before they attach. The next thing that we can talk about is personal preventive clothing. So Lee, as you mentioned, yes, you can tuck your pant legs into your socks. You wear long pants, long sleeves, put your hair up inside a hat if you're out, so that you're not providing anything that the ticks can easily grab onto. Additionally, wear light colored clothing so you can see ticks crawling on you. Also great if you're working with mosquitoes. And finally, and perhaps I would say the second most important thing, is using an EPA-approved insect repellent. And there are ones that you can use topically, such as DEET, IR3535, picaridin, and oil of lemon eucalyptus, which coincidentally is not the essential oil. It is an actual different thing. Catherine mentioned permethrin, which is used in mosquito spray. You can also treat your clothing or camping equipment. I like to treat my tents and my hammock with it. It's great. It lasts for several washes. You just have to be careful and always follow the instructions on the label when using a repellent product, because different ones work better in different situations and cause different biological effects. So.
Awesome. Thank you for that. That was very thorough, Megan. Appreciate it. And last but not least, John, I'm going to repeat the whole question because I know we've gone through a few. So what has been the most challenging aspect about communicating with the public and professionals about entomological triggers of allergies?
Well, I will say that I am fortunate in that most of my communication is one-on-one with people who have come to my clinic because they are having a problem of some kind. Just like with Megan, nobody really likes ticks, and they're probably, once they're aware of what to do about them, people are more eager to be rid of the ticks. Similarly for cockroaches, nobody actually wants a cockroach infestation, or at least I have yet to find a family that's like, oh, we kind of wanted to keep those. Now, it may be that the family had treated it more as a nuisance factor. They're irritating to have around. But once they understand that the cockroaches are actually driving the poor health outcomes from the kids' asthma, this is where they say, well, okay, now we're going to move this up our priority list. And families do have a lot of priorities that are on their minds. And then fortunately, because of the research that we've done here, we're really able to say that the use of insecticidal baits is very effective at eradicating cockroaches that are living within homes. So that conversation tends to go well. They recognize once they're told that the cockroaches are linked to the asthma, they say, "Great, I didn't want the cockroaches anyway." You talk about a fairly inexpensive and effective way of eradicating the cockroaches. They're really happy about that. Now, for the venom desensitization, it turns out nobody really wanted to get stung. But these are a lot of native species, or in the case of honeybees, this is an agricultural um, sort of species that are there and they're not going anywhere. So if you're allergic to honeybee venom, we're not going to do aerial spraying for honeybees to try to eradicate them. We're trying to keep them around. Likewise, for a lot of the paper wasps and yellow jackets, these are native species that are part of our ecosystem. So in those cases, it really isn't about controlling the insects, it's about controlling the immune system. This is where folks will come into me. We'll talk about the pros and cons of this approach, but ultimately we'll use purified venom extract to desensitize the patients. It's a long procedure. It's not without risks, but for nearly every patient, we're able to get them desensitized. So that in the case of beekeepers, they can continue keeping their bees. In the case of people who want to be out and about, where there are stinging insects that might induce anaphylaxis, they can go out and about. Though if they're stung, they won't have one of those life-threatening allergic reactions. Separately from interacting directly with patients, those conversations generally go well. It is interacting with other physicians. So most physicians are not entomologists, and they really use a one-size-fits-all approach to prevention of the interactions between different species. So talking about different cockroach species that have different health consequences, it's something we do through research presentations. Similarly, when we're talking about variable exposure to different types of stinging insects, this is communicated out through scientific presentations. And again, in general, goes pretty well.
Thank you,
Don.
Bob. This is amazing. Sending it back over to you, Dr. Michaels. Go ahead.
Thank you. I was just going to say, in the interest of time, I know we're getting close. Just wondering if there's any questions from the audience first.
Looks like we have two questions. Kayla, you want to get into them?
Yes, we can get into our questions. Thank you to our panelists. Thank you, Dr. Michaels, and thank you, Lee, for being here today for such a wonderful discussion. The very first question that we have here, um, and for all of my audience, please, if you have any questions for any of our panelists about any of the topics we have discussed thus far, please drop those in the Q&A and we can begin addressing some of those. So our very first question is, what were each of your first jobs after graduating and how did you get them? And we can answer that in any order in which you guys like, our lovely panelists.
Okay, I'll step up. So after graduating in 2007 with a PhD in vector-borne disease, I guess I started working as a lab manager for Dr. Don Wesson, whom everybody probably knows on this call. But then after that, my wife and I moved to Richmond, Virginia for her medical residency at Virginia Commonwealth University. And moving to a place that didn't have a public health school meant kind of trying to find your own niche and begging people. I worked for University of Richmond. I did adjunct teaching there in environmental studies. I finally found a postdoc with biostatistics department at Virginia Commonwealth University. I taught spatial epidemiology. You just try to find whatever niche when you end up in a place that doesn't have a real defined public health school. That's what I ended up doing.
Megan, we can go with you next.
Sure. So I touched a little on it. I moved to Maine. So while I was in my last semester of my master's program at Tulane, I was also actively applying to jobs and following a lot of job boards on the AMCA website, so American Mosquito Control Association, Ecolog, and others that tend to have vector-borne disease jobs associated with them. And I got the call. I'd never been to Maine, but the job looked pretty cool. I'd taken epidemiology. And I actually was in Florida at the FMEL Advanced Mosquito ID course when I had to take my first interview sitting in the Florida heat right about this time of year, in a little Honda Fit, answering questions with very limited cell reception. And, I moved there and I was lucky enough to enjoy where I was. And I strongly recommend networking as early as possible because the more people you know, the easier it is to find a job. So really start talking to your professors and your colleagues and looking at those internship opportunities that come up when they do, because you never know who might be able to help you then in the future.
And sorry, as shameless plug, Megan, you are hiring an intern this summer?
Yes, we do have an internship open this summer via the Pacific Southwest Center of Excellence as a partner. It'll be working on ticks in the high Sierras and looking at Colorado tick fever and Rocky Mountain spotted fever. So if you're interested, it is a paid internship, but you do have to be in California.
Megan, would you like me to drop your email address into the chat for anybody that might be interested or know someone?
Yeah,
Okay,
absolutely.
I'll do that.
All right, thank you, Megan. Throw the same question to Catherine.
Yes, I got really lucky. I was with Sarah at the time. I just sent a cold email to the director of the Florida Keys Mosquito Control District. It was during that dengue outbreak, and I asked if I could just come and help. And I didn't ask for a job. I just wanted to be a part of the action. And he invited me in for an interview for that PCR position. And that was it. It's been a great 16 years.
Thank you. John?
So I went the medical route, so it's probably different from the question's intention of what do you do after a master's degree.
Well, so the second question, we haven't gotten to it just yet about the master's degree. This first one is just what was your first job after graduating? So if you want to just talk a little bit about what you did, because you graduated with both an MD and PhD, yes?
Yeah, so after the medical school training, you go into a residency. So I did a research integrated residency in pediatrics. And then after that, there's usually a medical fellowship. So I did allergy and immunology, and then joined faculty.
Thank you. We have several students at our school and prospective students that join our MD and PH program and our MD PhD program. So this is right up the alley for several of our folks, especially that are interested in tropical medicine and infectious disease. And then Brian.
Yeah, so after Hurricane Katrina, we moved back to Western North Carolina. And so I was back and forth to New Orleans for a number of years. And I got really lucky. It was a little bit of serendipity. I'd reached out to Western Carolina University to see if there were adjunct teaching opportunities, met with faculty in the environmental health program, and they encouraged me to apply to a tenure track position without my PhD, which is sort of unheard of these days. And so I actually started my position mostly as a teaching faculty member, ABD, so all but dissertation, and then Western North Carolina is home for us, and so we've worked hard to make this work from a professional perspective and building our research program with lacrosse on top of being at a smaller institution.
Thank you for that, Brian. Our next question, for our panelists who graduated and started working after earning their master's degree specifically, what sorts of jobs and what forums, and Megan, you touched on this a little bit, does anybody have any additional recommendations for forums where our master's students specifically should be looking for job opportunities?
Yes, actually, and this I briefly mentioned because I mentioned that the internship at California Public Health is with the vector-borne disease section is through our Center of Excellence. There are multiple centers of excellence in vector-borne diseases. I do believe Tulane is part of the Southeastern Center. but on their websites and newsletters and job boards, they often have employment opportunities. And certainly for California, that includes job opportunities in our local vector control districts because we have one of the largest networks of mosquito and vector control districts in the country in California. So we work very closely with them. So I would say check out the Centers of Excellence.
That's great. Thank you so much, Megan. Shifting gears a little bit, we have another question. Can someone talk about what are some things you should do to prepare yourself to prevent getting bitten if you know you're going into an environment that has mosquitoes? And anyone can take this on the panel.
But Megan really wants to answer it.
I feel like Megan already answered it really with permethrin and EPA approved repellents, of course. These are all the things that we already really know. You got to cover up, you got to wear long sleeves, which is difficult to do in the heat and humidity. Limit your time outside at the peak biting times, those things. But certainly that list of EPA approved or EPA registered repellents is the first place to start.
Yeah, and I would also follow up with reapply. So if you're hot and you're sweating, you'll need to reapply the repellent more often.
Also, shameless plug, wear sunscreen.
Solid advice.
Well, that is perfect. That is great. Thank you so much. At this time, we don't have any additional questions. If any members of our audience would like to drop any last questions, that would be great. In the meantime, for my panelists or Dr. Michaels, if there's anything else that you would like to add at this time that you may have missed, or just some additional helpful tips and tricks for our audience, for folks who may want to get involved in different aspects of vector-borne control, please feel free to drop those now.
I'll just put in that it's really fun work. I mean, you don't run into anybody who doesn't love their job. It's just universally, people really like doing this kind of work. So if you're interested in it and you want to be a happy person, this is definitely the career track for you.
I concur. Like, I am working in higher ed now, but I worked in public health in this field, vector control, for over 10 years. And it was really fulfilling, being able to talk and meet residents and just tell them all the things that they may not know, to prevent things like Zika and West Nile virus from spreading. And it was just really fulfilling work. And so, yes, if you are interested in going into this field, do it. Like, it's fun. You get to be outdoors. You get to meet lots of interesting people. I enjoyed the work I did.
I'd like to add that while you're in graduate school, take the opportunity to skill up, to go after the things that maybe are outside your comfort zone. I guess is a hallmark of my presentation today. Maybe it's social science, maybe it's biostatistics, because man, that was a tough, I think I took that twice, but I got through. Go towards the things that you find difficult because they're going to make you better at it. And you really do need to find a way to differentiate yourself amongst everyone else applying for those same jobs. So that means for our line of work, it means GIS, it means statistics, those kind of skill-based things that may not be what drew you to public health, but those are the things that are going to help you going forward.
Thank you so much, everyone. That was some amazing advice and some good feedback. We do have one final question. We have some thanks in the chat, thanking everyone for their time today and presenting such amazing slides. But ultimately, aside from repellents, what are some other outdoor control methods for mosquitoes specifically?
Yeah, it's another tough question. So spatial repellents are very effective. I know that's not aside from a repellent, but not a topically applied repellent. So the Thermacell products work really well. What else to do for mosquitoes? Obviously, eliminate where they're coming from, as Catherine was mentioning, around your house, those containers and whatnot. But oftentimes they're flying in from many miles away. So that can be very challenging too. So that's where you have to rely on services like ours, public agencies that are doing community-wide mosquito abatement to try to limit those numbers. And if you have one, for instance, if you're in New Orleans, don't be afraid to use them. Don't be afraid to let them know you're experiencing more mosquitoes than you think is tolerable. That's what we're here to do. So it's tough on a backyard by backyard basis. Yes, there are insecticides you can use, and you can purchase at Home Depot to apply to bushes and whatnot that will have a residual activity against mosquitoes. But that's a little bit more aggressive of an approach as well.
Yeah, I agree, Kevin. I would never advocate for that if they know that we at FKMCD could be there the very next day if they just give us a call. So I'm not sure how to answer that question outside of my own district, but if you do have access to a mosquito control district, I would highly recommend giving them a call and telling them of your concerns.
All right. Thank you guys, and we are right at the top of the of the hour. So I want to thank all of our panelists, thank our attendees. Thank you all so much for joining us today for our Public Health Discovery Seminar. Thank you, Dr. Michaels. Thank you, Lee. And I hope that everyone has a wonderful rest of their day.
Thank you.
Bye, everyone. Thank you.